Hello everyone, for my third project of this semester, I will be explaining the details about the disease toxic shock syndrome. Toxic shock syndrome is a disease caused by the colonization of the bacteria Staphylococcus aureus or Streptococcus pyogenes, also known as group A strep. It's more commonly caused by S. aureus, so I will be focusing on the effects caused by that bacteria. It's often associated with the use of tampons, but it can be caused by other factors. In menstrual TSS, it is acquired by the use of superabsorbent tampons or by leaving a tampon in for longer than advised. It can also be caused by the use of menstrual cups or female barrier contraceptives. Non-menstrual TSS can be caused by Staphylococcus aureus infecting a wound, such as cuts, burns, or even bug bites. And it has been known to occur after childbirth and shockingly, or at least I found it shocking, when you pack your nose to stop a nosebleed. Toxic shock syndrome is an infectious disease caused by an overgrowth of Staphylococcus aureus, but it will not be spread from person to person unless by direct contact. In the United States, for every 100,000 persons, only around 1 to 3 people will acquire toxic shock syndrome. As this is a severe disease, the death rate can be up to 60%, potentially higher if left untreated. This disease's prevalence spiked in the 70s all the way up into the 90s because tampon production was much less regulated and not much was known yet about the disease or the risk of tampons. Menstrual toxic shock syndrome is the most commonly acquired form of toxic shock, so this disease does affect more women than it does men. People who are at a higher risk are those who frequently wear tampons or wear tampons for long durations, people who are younger, older, or immunocompromised. To the right, I have included an image that depicts the number of cases of toxic shock syndrome in relation to the age of women who acquire toxic shock syndrome compared to the number of cases of women who regularly wear tampons. As you can see, the most frequent age group to acquire toxic shock syndrome in both categories is 15 through 19. And in all age groups, the women who regularly wear tampons have a higher rate of acquiring toxic shock syndrome. Symptoms of toxic shock syndrome will start with flu-like symptoms like headaches, body aches, and fever, but it can quickly develop into way worse symptoms. Hypotension or low blood pressure is a common symptom of toxic shock syndrome, which can lead to fainting and fatigue. Characteristic signs of toxic shock syndrome include redness of the eyes, mouth, and vagina caused by an increased blood flow to those areas. In individuals with dark skin, strawberry tongue is a symptom caused by the increased blood flow to the mouth, as shown in the bottom left image. Often, a red rash or peeling skin on the arms and hands will form that resembles a bad sunburn. As symptoms worsen, confusion, seizures, and organ failure of the kidneys, liver, and/or heart will occur. Diagnosing toxic shock syndrome can often be very difficult because toxic shock syndrome resembles several other diseases like Rocky Mountain spotted fever, caused by rickettsia, or toxic shock-like syndrome caused by Streptococcus pyogenes. In order to rule out these other diseases, bacterial samples are taken by swabbing the vagina, cervix, or wound, and then they are cultured to determine what bacteria is causing these symptoms. They will determine which bacteria is colonized by performing a series of chemical testing and viewing the bacteria in a gram stain, which I will explain a little bit more on in the next slide. Often, blood will be drawn and viewed as a peripheral blood smear because often patients will have thrombocytopenia, which is a decreased platelet count. They'll also have anemia, which is a decreased red blood cell count, and leukocytosis, which is an increased white blood cell count. Samples of urine or spinal fluid collected by a spinal tap will be drawn to determine if the infection has spread systemically into the blood, kidneys, or spinal fluid, causing the patient to become septic. Staphylococcus aureus is a very common bacteria that is usually harmless and is even found in the natural bacterial flora of the skin in many individuals, but problems can occur when it colonizes in areas that it shouldn't. Toxic shock syndrome is caused by specific toxin-producing strains of Staphylococcus aureus, and that can actually include MRSA, which is a well-known dangerous strain that is resistant to the antibiotic methicillin. S. aureus is differentiated from other bacteria by performing a gram stain along with other testing. In a gram stain of Staphylococcus aureus, it will appear as a purple gram-positive coccus due to its thick peptidoglycan cell wall. Staphylococcus aureus tests positive for both enzymes catalase and coagulase. Coagulase is an enzyme that will coagulate blood plasma, and it is tested for by mixing the bacteria with rabbit plasma. If positive, the plasma will form clumps. Catalase is an enzyme that neutralizes the bactericidal or bacteria-killing effects of hydrogen peroxide. It is tested for by adding hydrogen peroxide to a bacterial sample. If positive, the hydrogen peroxide will begin to fizz and bubble. Staphylococcus aureus is a halotolerant bacteria, meaning it can grow in salty environments such as the human skin. This allows for it to be grown on mannitol salt agar. Unlike some halotolerant bacteria, Staphylococcus aureus can ferment the carbohydrate mannitol, which causes the agar to turn from red to yellow. This is shown in the top image that compares Staphylococcus epidermidis on the red half compared to Staphylococcus aureus on the yellow half. When grown on blood agar plates, Staphylococcus aureus displays beta hemolytic activity. This means that it is able to completely lyse red blood cells. In the bottom image, you can see how around the colonies of S. aureus, the red color from the blood agar has turned into a clear yellow color. Toxic shock syndrome affects multiple systems in the body, but I have chosen to focus on its effects on the adaptive immune system. A healthy immune system has two components, the innate and the adaptive immune response. The innate immune system is known as the first line of defense. It is a fast-acting general response to any pathogen or foreign body. The innate immune cells, such as macrophages and dendritic cells, will then go on to trigger the adaptive immune response. Adaptive immunity is much slower to enact, but it has a specific and destructive response to antigens. Because the adaptive immunity is a destructive response, adaptive cells must be able to differentiate between self-antigens and non-self-antigens. It does this with the use of self-receptors that are specific to oneself. Without this feature, a dangerous autoimmune response will occur. Adaptive immune cells begin as a lymphoid progenitor cell, but then will mature into either T lymphocytes or B lymphocytes. B and T cells will further differentiate into different cells with a specific response to antigens. B cells will differentiate into either plasma cells, which secrete specific antibodies for an antigen, or memory B cells, which are able to remember the antigen and the response to that antigen. T cells can mature into helper T cells, cytotoxic T cells, regulatory T cells, or memory T cells. Memory T cells have a similar function to memory B cells, allowing memory of antigens, allowing for an efficient response. Cytotoxic T cells, or CD8+ cells, will kill foreign cells like bacteria, cancerous cells, or infected cells. Regulatory T cells maintain homeostasis by suppressing an immune response. Helper T cells, or CD4+ cells, do not have a destructive response, but rather they will stimulate other immune cells like macrophages, B cells, and CD8+ cells, causing them to rush to the area of infection. These cells are stimulated by the specific cytokines that are released by CD4+ cells. Before a CD4+ cell can release these cytokines, it must first be activated. CD4+ will become activated when an antigen is presented by an antigen-presenting cell, or an APC, such as B cells, dendrites, and macrophages. The antigen is presented on the major histocompatibility complex class II, found on the external membrane of APCs. In the image, you can see a dendrite, shown in yellow, presenting an antigen to the CD4+ cell, shown in blue. The CD4+ cell will then release cytokines that will stimulate another immune cell in this case a CD8 plus cell shown in green. As I said previously, the strains of Staphylococcus aureus that cause toxic shock syndrome produce toxins. These toxins are also called superantigens. These superantigens are able to penetrate through endothelial cells of mucosal membranes like the vagina or the nose, or into the site of a wound. Once inside the body, these toxins can cause negative effects throughout the body. Staphylococcal superantigens require an increased amount of oxygen in order to be released, and it's theorized that toxic shock syndrome is frequently caused by tampon use because the insertion and removal of tampons increases the partial pressure of oxygen inside the vagina. Normally, APCs will present the antigen on the MHC class II to the receptor on CD4 plus cells, but staphylococcal toxins will bind externally to the side of the MHC class II complex and the T cell receptor, as shown in the image to the right. Normally, only 1 per 10,000 T cells will be activated in response to an antigen, but the binding of the toxin to the MHC complex and the TCR will cause up to 20% of the T cells to become activated. This causes a very large amount of cytokines to be released. This is also known as a cytokine storm. This overproduction of cytokines are what causes the symptoms of toxic shock syndrome. To dig a little deeper into the biochemistry and molecular biology of the MHC class II complex, it is a heterodimer composed of an alpha chain and a beta chain that are non-covalently bonded together. Each chain of the MHC class II complex has 2 domains. The domain alpha 1 and beta 1 form the binding cleft where the antigen is bound to and then presented to the CD4 plus cell. The alpha 2 and beta 2 domain form the immunoglobulin-like domain that contains receptors and functions in cell recognition. The transmembrane segment of the MHC class II complex is composed of hydrophobic amino acids, and it acts as an anchor, allowing it to be stably secured onto the antigen-presenting cell. The major histocompatibility complex is also referred to as a human leukocyte antigen, or HLA, because they were first discovered on a leukocyte. The image on the right shows both a cartoon image and a ribbon model of the structure of the MHC class II. You are able to see that the 2 chains are composed of 2 domains along with the peptide binding groove. CD4 plus cells are unable to bind to and respond to free-floating antigens. The antigen must first be processed and then displayed by an APC to elicit a response. The T cell receptor, or TCR, is a heterodimer that contains an alpha chain and a beta chain that are stabilized by a disulfide bond at the base. Each chain contains a variable region and a constant region, as shown in the top image. The variable domain of the alpha and beta chains are where the antigen that is presented by the MHC class II complex is able to bind. This bond between the TCR and MHC complex is stabilized by another T cell receptor called CD4. The bottom image shows the ribbon model of the TCR. The alpha chain is shown in blue and the beta chain is shown in green, with the transmembrane segment at the bottom with all of the alpha helices. Before I get into the biochemistry of staphylococcal toxins, I want to explain the differences and similarities of exotoxins and enterotoxins that are both produced by S. aureus. Both exotoxins and enterotoxins are proteins that contain a catalytic domain and a receptor binding domain. The catalytic domain of these toxins are what cause the toxins to have negative effects. Exotoxins and enterotoxins are both produced solely by gram-positive cells, such as S. aureus. Exotoxins are released from a cell into its environment, hence the prefix exo. Exotoxins are sensitive to heat and will be killed or inactivated at high temperatures. Exotoxins trigger severe effects throughout the body by disrupting the healthy metabolism of the cell or even just lysing the cell. Enterotoxins are a specific type of exotoxin that affect the digestive system. A disulfide bond is found in enterotoxins to stabilize their tertiary structure when in the digestive tract. Unlike with exotoxins, enterotoxins are able to withstand higher temperatures. The cytotoxic effect of these toxins will alter the ion permeability of epithelial cells in the digestive system. Enterotoxins are able to form pores in membranes that cause the permeability of chloride ions to increase. This increases the movement of sodium ions and water into the lumen, giving rise to symptoms of nausea, vomiting, and diarrhea. The most common cause of the effects from menstrual toxic shock syndrome is from the exotoxin toxic shock syndrome toxin 1, or TSST1, which is produced by S. aureus. TSST1 is a monomer that is composed of 234 amino acids. The bottom image shows a ribbon model of the structure of TSST1. TSST1 is a toxin produced by phylogenetic group 1 strains of S. aureus. This toxin functions by binding to the MHC class II alpha or beta chains and the variable region of the beta chain on TCR, as shown in the upper image. When bound to the MHC class II complex and TCR, it skips the antigen processing step, triggering many CD4+ cells to become activated. Once activated, CD4+ cells will secrete copious amounts of cytokines such as tumor necrosis factor alpha, interleukin-1 beta, and interleukin-6. The mass amount of these cytokines lead to symptoms such as fever, inflammation, severe tissue damage, and shock, which can lead to death. Another common cause of toxic shock syndrome, particularly non-menstrual toxic shock syndrome, is from staphylococcal enterotoxin B, or SEB. SEB is not quite as toxic as TSST1, but it will still cause severe damage to the patient once it is spread throughout the body. SEB is a monomer composed of 265 amino acids that is produced by S. aureus strains in the phylogenetic group 2. The bottom image shows a ribbon model of the structure of SEB. Similarly to TSST1, SEB binds to either the alpha or beta chain of MHC class II and the variable region of the beta chain of TCR, leading to a cytokine storm produced by CD4+ cells. In the top image, you can see how SEB will bind to the external portion of MHC class II, away from the antigen binding cleft. The CD4+ cells will secrete a large quantity of cytokines, including tumor necrosis factor alpha, interleukins 1, 2, 4, and 6, and interferon gamma. An increased amount of these cytokines causes symptoms of nausea, vomiting, and more severe symptoms like organ and tissue damage. Very rarely is toxic shock syndrome caused by staphylococcal enterotoxin C, but it is not unheard of. SEC is a monomeric protein composed of 266 amino acids shown in the image to the right. SEC has 3 different subtypes that it can be produced as: SEC1, SEC2, and SEC3. The image on the right is the ribbon model of the SEC type 2. Strains of S. aureus that produce SEC are from the phylogenetic group 2. Like with previous toxins, SEC binds to the MHC2 complex and TCR, but it can only bind to the alpha chain of the MHC class II complex. This is because, unlike all other toxins produced by S. aureus, it lacks the zinc binding domain at the C-terminal, which is required to bind to the beta chain of the MHC class II. Despite being able to bind to only one chain, it still triggers a massive cytokine storm by activating many CD4+ cells. The cytokines released by CD4+ include interleukin-2, tumor necrosis factor, and interferon gamma, causing symptoms of inflammation, rashes, along with nausea, vomiting, diarrhea, and organ damage. To treat toxic shock syndrome, first antibiotics are given. Usually nafcillin or oxacillin are given intravenously. However, in patients with a penicillin allergy, vancomycin can be given as a substitute. The antibiotics used to treat toxic shock syndrome must be beta-lactamase resistant in order to prevent the beta-lactamase of S. aureus from inactivating the antibiotic. Clindamycin is often used first few days in conjunction with other antibiotics because it halts the production of toxins. Another treatment for toxic shock syndrome can be with the use of pooled immunoglobulin from healthy donated blood. Once purified, it is injected into the bloodstream to help the patient's body fight off the infection even further. Often, oxygen is given to the patient to support their breathing, and fluids are given to reverse hypotension by increasing blood volume and to prevent organ damage. In severe cases, dialysis might be necessary in patients with kidney failure, and amputation might be required in patients who have suffered from gangrene due to loss of blood flow. As it is Women's History Month, I would like to spotlight Lauren Wasser, a survivor of toxic shock syndrome. In 2012, at the age of 24, she contracted a severe case of toxic shock syndrome. She suffered from organ failure and had to amputate both of her legs below the knee. Now she advocates for the production of better and safer tampons so this disease stops affecting other women and girls. She wants all menstruating individuals to be aware of the risks that are associated with tampon use. To summarize what you have learned in this presentation, I would like you to draw to the best of your abilities, it doesn't have to be perfect, the complex formed between the MHC class 2 of an APC, the TCR of a CD4 plus cell, and a staphylococcal toxin of your choosing. On the image to the left, drawn by yours truly, I drew the antigen-presenting cell in red with the MHC class 2 in green. I drew an antigen in purple on the MHC class 2, and then I drew a CD4 plus cell in blue with the TCR drawn in orange. In the center between the MHC class 2 and the TCR, I drew the toxin in pink. These are all of my citations that I used on this project, and I hope you enjoyed. Thank you so much.